So you've been hearing about non-RCTs so far, right? And there's a lot of questions were related to dealing with confounders in this. And this is one of the main problems we have in observational studies that we tend to overcome in clinical trials, but only if we randomize, and second, only if we randomize a lot of people. People think that, oh, if I do a randomized controlled trial and I have 100 patients and I randomize 50 to get intervention A and 50 to get intervention B, I randomize everything is fine. That is a misconception. Randomization will allow you to do one thing, which is to say, I have the statistical ability to be able to look at the group A versus group B because I have not assigned people in maybe a biased way, but I've done it objectively. But there's two things that randomization is supposed to do. One is to do the objective assignment to intervention, but the second one is to deal with these confounders. And if you randomize only 100 people, it's not going to work. I can almost guarantee you, you will not get perfectly balanced things. People try to do these things in cohort studies and try to pretend you have a real trial because people have been doing something here and you have some people that are exposed and some people that are not exposed, but they're not comparable. And you try to do this propensity score matching to create groups that are comparable. But that's only on things that you know. And it's not on things that you do not know. So we have to be very careful with this. So design issues are very important. So we'll be talking about different types of designs, and we'll also then, after I think the break, we'll talk about the statistical hypothesis. So many of you are familiar with the what are called the phases of drug development. And there's this thing called phase one, which is when they're taking a drug has already gotten past the preclinical stage, which is where it could be in vitro, it could be animal studies, etc. And whatever medication it is, has now said, okay, let's start using it in humans. And phase one is the first time they use it in humans. They call them trials. I don't call them trials. A trial for me needs to have a comparison arm and it needs to have a randomization process. Phase one is not. This is just finding out, we give it to a few humans, pay them a lot to volunteer to see if they take this thing that no other human has ever taken. Will they die? Will they get something atrocious? We hope they don't, but that's it. And that's just to get toxicity, to find out if it's toxic or not. If it passes that, now they want to do a comparison one, and there'll be phase two. But phase two sometimes will be initially looking at what is the proper dose that we should be given. And they have all of these dose finding studies, and they will try multiple doses and start going up and dose until people don't tolerate it anymore. And again, there's no randomization here. Sometimes they will do some randomization to those different doses, but otherwise they will be giving the same dose and incrementing in a given person. So some of the first phase two or 2A, they call them, the dose finding ones, are not really trials either, right? But then eventually they get to 2B, and 2B is now, we're going to randomize some people to this dose versus the other comparison arm, whichever it is, and see the safety of it and efficacy in it. And we'll do it in a controlled situation. So they're looking at it in small numbers of people, 150 to 350 or so. They are carefully selected. They are homogenized, standardized. So they'll try to eliminate a lot of other potential confounders. Oh yeah, we're going to study, men between 30 to 39 with this particular condition that don't have that, that don't have that, that don't have that. And therefore, it's a very controlled situation. We are going to ensure they take the medication. And so we are looking at if it's is it going to work or not? And then what we call the phase 3 trials are what we understand as the main trials that we're going to be talking about in this course. We're going to talk a little bit about phase 2, and somebody will talk about phase 1 later. But phase 3 are the main, what people think are clinical trials, which are usually large number of people. There's a little bit more diversity in these people, and we're looking at safety and efficacy in them. And this is what we'll talk about. Once a drug gets approved, usually then they have what's called a post-approval studies. The pharmaceutical industry calls them trials. They're not trials either. This is just monitoring what is happening in the community once a drug gets approved. Because when the drug is being tested, it's being tested in a very controlled situation where people are supposed to follow a protocol, and the physicians and the clinical staff, medical staff, is also following a protocol on how things should be done, how the intervention should be done, and in certain types of people that meet the eligibility criteria for the study. Once a drug gets approved and it's out there in the market, God knows who takes it, who gets it prescribed. Physicians may start prescribing it to people that were not studied. If it worked for men, the study was for men, now they'll prescribe it to women also. If they did it in older people, oh yeah, younger people too, or very old people too. Oh, they didn't use any, they excluded everybody with renal disease, but we'll also give it to this one. And the people taking it. Now, how many of you have had to take antibiotics, and usually you have to have a 14-day period. You get prescribed an antibiotic by your physician. What do? Oh, my aunt had some. I'll just use hers, not this new one. And then I'll go, after 3 days, I feel okay, then I stop. Or after 10 days, I'm still not feeling okay, I go and I buy something else. People do all sorts of stuff, and they're not following any protocol. That's real life. And the pharmaceutical companies are interested in seeing what is happening with my medication in real life. It's not a trial, that's a monitoring, very much like this quality improvement thing, which was in a factory or it was in a particular clinic. Now this is in a community. It's almost like looking at that. So I want to be clear on the definitions. When we're talking about clinical trials, let's not mix all sorts of stuff. We're talking about randomized ones where you're comparing A to B, and we'll have other designs. And we're looking at either a small number or a large number of people. All right. We've already seen this. We want to establish evidence, and we've already seen this, so I'm not going to go through this again. But I want to mention why trials are so important. And that is because if you do a trial well, and you do a trial that convinces people, that's what is a successful trial. It's not whether you get a p-value that's significant, it's whether it's done well. But this is historically, I was lucky when I finished my postgraduate education. I went to work at this group that had started a trial when I was in high school. They started in 1971, I was in high school. And so I had nothing to do with this trial, except I came in at the end of the trial. I finished in 1980, so I worked for 2 years on this trial. And I was lucky enough to be involved in this trial that was the definitive study that established cholesterol as a risk factor in people, in men 35 to 59 years of age with high levels of cholesterol. There had been a lot of evidence from doing case series that people that died from heart disease had high levels of cholesterol. There have been many studies on case control studies that had shown that there was a strong association. There was even the Framingham study that had been going on that was a cohort study that showed that people that had high cholesterol had more risk of dying of heart disease and heart attacks, but people were not convinced. Who was not convinced? People that had economic interests in it, the people that made butter and cheese and the eggs and the bacon and the meats, and all of these people said, no, cholesterol, unless you show me that if you lower the cholesterol in people that have high cholesterol, and it does something, it causes a reduction in stuff, that's the only proof that will convince us. And so in that time, in the 1960s and 70s, it was the major #1 cause of death in the United States. So the government decided, we'll invest money in it. They invested $120 million back in those days. That money today probably would be $1 billion because of the inflation. There was a lot of money in those days. They had 12 centers. They had terrible interventions because that's what was available at the time. Cholestyramine was, many of you are too young to know, was a horrible sawdust thing that you had to mix with water or orange juice and take it 6 times a day. And the pharmacoepidemiologist said to get an effect from this, you have to take it for 7 years minimum. Garbage sawdust. In fact, I got to try it. It's horrible, disgusting. And they gave people low cholesterol diet as well. Why? Because there was enough evidence that low cholesterol is a good thing. It was unethical to do placebo alone. So the control arm was placebo pill, or placebo, not pill, placebo sawdust. Imagine how horrible it was. The sawdust causes diarrhea and constipation. And in order for people not to get unblinded, the placebo also, they put some ingredients in it to cause GI problems. So imagine taking this horrible thing 6 times a day for 7 years. They convinced 3,800 men to do this. They did it, and what they showed was that it worked. And therefore, they got, for I think every 1% drop in total cholesterol, they got a 2% drop in the risk of heart disease. And this was such that came out in 1983. It made the cover of Time magazine. But Time magazine, what did they do? Everybody should worry about cholesterol. Men, women, children, elders. They never studied women. They never studied children. They never studied older men. Why do they do that? Because this is what sells Time magazine. This is what sells newspapers. So be careful when you do a trial. We are also very, very careful about who the study results apply to. And we'll be talking about the study of the trials results only apply to the people you study, the type of people you study. And the inclusion and exclusion criteria is very important. All right. So statistical reasons the medical community loves RCT. Everything is complex. God made the world very complex. We simplify models. The RCT is a simplified model to test one thing. We have confounding everywhere, and we have all of this confounders may affect what we're observing. So experimental research design, control factors by design. And this is what, we like to do. And then maybe if there's some residual confounding, you may do it by analysis. But observational research designs, you cannot control many factors. You may control some factors and say, I want to study smokers versus non-smokers. But do you control how much they smoke? Do you control how much they don't smoke? You cannot really do it because you're just observing. In these studies, you would be able to control it. This is why the tobacco industry still says there is no evidence that smoking causes emphysema or cancer or heart attacks. Why? Because there isn't a study where we say, okay, you are randomized to smoke 40 cigarettes a day. You're randomized to not smoke 40 cigarettes a day. Let's follow you and let's see what happens. It's unethical to do. And because it's unethical, there isn't a trial. And so the powers that be, the tobacco industry says, oh, there is no cause and effect until you show me. This is the problem, right? So why the medical community loves it is because you are in control. When you are doing a trial, you are the one that controls everything. When we talk about design and the operational parts this afternoon, you will see that you, as the investigator, you decide everything about it. You decide the intervention. You decide what is the comparison. You decide how to measure it, who to include, how long to follow, how much to do. And so when you control it, you're doing what we've all been trained to do when we were in high school and we took chemistry lab or biology lab or physics lab. What did we do in the lab? We did real experiments, right? We took a bunch of reagents. We knew which reagents, we knew which concentration, we knew the temperature, we knew the pressure. And what did we do? We changed one thing. And then we saw what happened. We had a cause and effect. We raised the temperature, or we applied more pressure, or we added something. This is what we're trying to do in a controlled trial. We are wanting to have everything like a true laboratory. The problem is we're dealing with humans. That's why animal studies, we can put them in little cages and we can feed them exactly what we want them to feed. We can't do that with humans. And this is the issue. We have this variability. In biology, we have a lot of more variability than in chemistry and physics. So we have biological variation, we have genetic variation, but we have behavioral variation. And so studies in ophthalmology where I'm just surgical trials are done in very few people because there is very little variation because on the surgery technique. Studies that involve taking a pill have some variation because it's the behavior of taking the variation. Studies and the trials apply to educational campaigns. They apply to exercise, changing your diet. Those are the hardest things to do because it involves people changing their behavior. And most people don't like to change their behavior, especially if you tell them you have to do it this way. How many of us have started a diet or an exercise routine and never followed it? So this is an issue. So you try to be in control. That's why people like clinical trials. So we want to control as much as possible because as this Dutch philosopher may have said it, but as every mother says, it's better to prevent than to treat and to deal with it. So let's try to prevent things. So we prevent those confounders from creeping in by design. And try to control as much as possible is the way to do it. So controlling by design is restrict. How do we do it? We restrict the study population. We don't study everybody, young and old. We study people that are that have the condition, but we homogenize them as much as possible by my inclusion criteria. Who decides the inclusion criteria? You as the investigator. And you eliminate those people that have other potential problems. This is a good thing because it homogenizes, eliminates variability or variation or heterogeneity, but it also says, well, the results only apply to these people that I studied. It doesn't apply to everybody else. We standardize the intervention and we also standardize the control. Even some studies that say, okay, we're going to do this intervention A versus usual care. Usual care is controlled as well. It's not usual care, whatever people want to do. No, usual care following these standards. And so it is always controlled as well. So you standardize the intervention and you standardize the control and you standardize how you implement it and you monitor. If you don't monitor how people are following the protocol, I can guarantee you that the staff is not always going to follow the protocol. People that are investigators tend to blame the participants of the study for not adhering to the interventions They forget that their staff are also human beings, and no human being likes to be told exactly how to do things. Yes, they will do it for a job, and they will also, but keep in mind they're humans, and every human will look for a shortcut. Some humans will look more for a shortcut than others, and they will not follow the protocol. I can give you some examples later on that will amaze you of what people do. So you need to monitor compliance and adherence. And then we vary only one factor, right? That's who gets the intervention versus who gets the other. So this is why we like it, because we can control things. Now, we end up doing other types of analysis that are not the primary analysis. And we study, if we don't get the exact balance that we want, because we didn't randomize a lot of people, we may start looking at some subgroup analysis. And many times we even look at subgroup analysis for looking at consistency of results in smaller groups. And so we study more homogeneous subgroups of the sample, and just say, okay, let me just study the men in the sample. Let me just study the women, and do I get consistency of it? And we adjust the effect of potential confounders that were not totally controlled for, because I did not do a very large trial. So we have different types of trials, and these are done in such a controlled fashion that we end up with trials that are, as I said, not really applicable to the general situation. Not every hospital is going to have the resources that CMC Vellore has, or that Ames has, for example, to do it. So once you show that something works, is it going to work in every other hospital, in some rural hospital, in some place that doesn't have these resources, or doesn't have the trained investigators and physicians to do it? So what we should do is, is the protocol so hard to follow that they won't be able to follow? So people are now talking about, especially governments that are funding this, WHO, et cetera, that why should we fund your trial that is going to study this thing that's going to benefit a very, very small proportion of people with that condition? Why should we invest that money? And so they're wanting things that are more applicable to more people in more settings, in more situations, okay, with the same condition. So we're talking now today, recently, about pragmatic trials. And a tightly controlled RCT addresses efficacy rather than effectiveness. And so it's when people follow exactly the protocol, do you get this to work or not? And what is preferable? Usually, we would like it to apply to more people. So we want it to be effective, even if it's not in the ideal situation. So efficacy is, does a treatment work under ideal, highly controlled, optimal conditions? Versus effectiveness, does a treatment work in real life clinical situations where, for a variety of reasons, it doesn't matter what reason there is, treatment application and adherence is not going to be ideal because we don't have the resources, or the people just don't follow things, right? And so efficacy trials have high internal validity, but don't have high external validity, while the effectiveness ones have higher external validity, but you still hope that they have internal validity, and you will get that hopefully if you do a randomization and you monitor things properly. So this is an idea that was proposed maybe about 14 years ago, and that is it actually, the word PROCESE is it's an acronym. People love acronyms, but they made it. It's a pragmatic, PR, explanatory, continuum, indicator, summary. But it's also what the Swedish language has as an accuracy word. And so what it says is, you should look at your trial and judge the design of your trial based on how much does it require for flexibility of the experimental intervention. How rigorous is that requirement? Or the comparison intervention. How flexible is that? Can it be? Is it so rigorous that it cannot be flexible? So you have this, the explanatory center is here, and you have, as you go out, you have increased flexibility, and you're more rigorous in the center, you're more flexible here. How much expertise do you require in the experimental intervention or expertise in the comparison one? Do you require a lot or you require less expertise? So you're more flexible as you go outside. You look at the follow-up intensity, the eligibility criteria. Who are you excluding a lot of people or not? And then what are your outcomes? Are they ones that are easy to obtain or hard to obtain? How much compliance do you need from the participant and the practitioner? And how much is, how rigorous you are in your statistical method for primary analysis? The more flexible you are, the more outside you are. And so they propose that studies that are very flexible will look like this, where you put a point out here and then you join the points. And if you're very inflexible, your points are closer to the center and you join them and it looks like this. And we all know that if we try to turn a wheel, it's easier to turn a wheel that's big than to try to turn it from the center, right? And so this one is going to be, their analogy is something that looks like this is going to be one that is more pragmatic and is going to be more useful because it will move things. A wheel like this moves things. A wheel like this is hard to move, right? So it's less pragmatic. It may be fantastic as a trial, but it's not as useful to society as this one. Now, interestingly, they look at some other trials, and not every trial is going to be flexible on everything or inflexible on everything. And you have some things that are flexible, and you just have a junky road when you're going on that wheel. So, we want pragmatic trials, and we want them to be good. And Dr. Tumbo is going to be talking about meta-analysis and systematic reviews as helping us synthesize that information. There's other types of ways of putting these together, and we may have time to talk about this meta-regression when we're doing systematic reviews and meta-analysis. If there is confounding still, because you're putting together multiple studies that do not control all the confounding by design, you want to do meta-regression. So there's different types of study designs on that, but we're not going to talk so much about that. RCTs, as I said already in my introductory joke thing, is that people think of it as a religion, and you should not. People tend to discount other kinds of evidence. And other kinds of evidence can be strong and important and useful. If you're able to convince the people you want to convince using other designs, that's fantastic. You don't need to do a trial unless people are not convinced, right? Be careful about that. Alternative evidence can come from these quasi-experimental designs and the observations. Okay. Questions, comments?